Hello, I'm Riti Sharan, Assistant Professor from Texas Biomedical Research Institute. On behalf of our collaborative team, I'm excited to share the recent work from my lab titled Modulation of Pulmonary IL-21 Expression during LTBI and MTB-SIV co-infection with you all today. In this paper, we present three major points. First, IL-21 positive, CD4 positive T cells are selectively impaired in lungs during MTB-SIV co-infection. Second, this defect persists despite treatment. And third, spatial transcriptomics reveals a persistent Th17 STAT3 biased pulmonary immune state and incomplete immune reconstitution. We are a multidisciplinary team from Southwest National Primate Research Center, Tulane University, and Emory University, studying immune dysregulation in TB-HIV co-infection using a non-human primate model. Chromogenic staining of tissue sections was performed by the lead author of the paper, Dr. Vinay Shivanna, who's a board-certified veterinary pathologist. Xenium data generation and analysis was carried out by our collaborators at Tulane University, and the histopathological assessment was conducted by investigators at the Southwest National Primate Research Center. And in addition, we had a number of authors who contributed to the writing, data analysis, and interpretation during this manuscript. The problem is that TB can remain latent for years, but HIV co-infection disrupts this immune control, leading to reactivation of TB. However, when immune restoration fails in the lung, even after therapy, and what leads to this is not well understood. From our previous work, we are aware that antiretroviral therapy is able to suppress HIV, but it fails to fully restore the MTB-specific CD4 T cell immunity in the lungs. We are interested in studying IL-21 because it's a cytokine that is critical for Th1, Th17 responses and is really unexplored in the co-infection space. And no prior NHP studies have been conducted to understand how do the bugs modulate this response during co-infection. So in this study, we aimed to define how IL-21, a key immune regulatory cytokine, is altered in the lungs during TB and SIV co-infection, and whether standard therapies can restore its function. So for this, we used our well-established macaque model to study TB and HIV co-infection, where we applied both antiretroviral and anti-TB therapies, combined them with advanced spatial and imaging technologies. We had 4 groups: low-dose TB infection only, low-dose TB infection plus SIV without treatment, low-dose TB infection plus SIV with just antiretroviral therapy, and the last group was low-dose TB infection plus SIV plus antiretroviral therapy and anti-TB therapy, which is the isoniazid and rifampicin treatment. We collected the tissues from these animals at necropsy, and we performed the chromogenic multiplex staining as well as spatial transcriptomics on them. The key discovery, or the first major finding, was that IL-21 positive CD4 positive T cells were very readily detectable during latent TB infection at 6 to 7%, but were profoundly reduced following SIV co-infection to 0.2 to 0.5%. And even though treatment with ART or ART plus 3HP improved the clinical outcomes and the bacterial control, the pulmonary IL-21 producing CD4 T cells remained relatively low compared to LTBI levels. The second finding was that this deficit appeared compartment-specific. So while treatment was able to restore the IL-21 levels in the colon as well as systemically, the pulmonary IL-21 producing CD4 T cells remained persistently impaired despite treatment. We went ahead and performed linear regression analysis, and we were able to show that IL-21 positive CD4 positive T cells frequency was able to predict the lung bacterial burden, or there was a correlation, but this correlation was not seen with just CD4 positive T cells. Further, the third finding, spatial transcriptomic analysis revealed that there was a shift from a balanced immune landscape during latent TB infection towards a STAT3 associated inflammatory state during co-infection, and then treatment partially corrected this imbalance but failed to fully restore the IL-21 associated pathways, and the balance was still tipped more towards the inflammatory STAT3 type of response. And so in conclusion, we know that there is a selective and a persistent IL-21 deficit that treatment does not really resolve. This deficit appears to be compartment-specific, as it is regulated to some extent systemically and in the colon. Further, there is a Th1-Th17 imbalance that is shown in the coinfection that treatment is not able to balance out. And so we think that exogenous IL-21 could reinforce the Th1 responses and restore Th1-Th17 balance. So our findings suggest that IL-21 associated pathways may represent promising targets for host-directed therapies aimed at improving the pulmonary immune reconstitution and preventing TB reactivation in HIV-infected individuals. As the senior author, I would like to thank all the members of our collaborative team, our funding agencies, and the staff of Southwest National Primate Research Center. And I thank you all for your interest in our work.